Good afternoon. My name is Florence Fontaine, and I would like to offer to you an insight into the philosophy of a cost action, and specifically one action that started in 2013 and will conclude this year on GTDs. The philosophy of a cost action is that of bringing together human resources and knowledge. We have 120 experts participating in the action. These 120 people come from 22 countries in Europe and also Algeria, Montenegro, and Israel and South Africa. And the participants in this cost action focus on different topics, including all GTDs. We have microbiologists, mycologists, experts in plant pathology, chemists, biochemists, plant physiology experts, epidemiologists, people who do disease management, and cultural and farmers. And then a cost action can involve all these categories, including nurseries, that can be private businesses and those who are in charge in private businesses of biocontrol. You saw the countries. The goal of the cost action was that of exchange information along four lines: pathogens and epidemiology, and then the goal there was that of having more insight into fungi and describing the disease spread in various European countries, and also understanding the contribution by the environment to the spreading of the machine. Oftentimes, we also speak of microbial ecology of fungi. And indeed, in a grapevine plant, we have an ecology. We have pathogens responsible for GTDs, but also a microflora, which is beneficial for the plant. The point, therefore, is studying that microflora, both the beneficial one and the pathogenic one, and see why a number of plants do not express the disease even though they have the pathogens in the wood, while others do express the pathology even though they have a beneficial microflora. And the third group then is the interaction between vine and pathogens and the expression of the disease. We want to understand the dialogue between pathogens and the plants in order to find the ways to contrast these interactions. And of course, these three axes aim at gaining knowledge on the topic and then fight the problem. This has to happen at nursery level because the pathogens are already present in nurseries, and this entails also working in the vineyards so that the entire sector and chain are covered. The goal being that of preserving vineyards in the long term. Now, I would like to describe the various actions and what we have done so far. Pathogens. Well, characterization and identification. First, in European countries, we set up a library of the main species of pathogens in Algeria, France, Spain, Italy, Germany. Of course, in all these countries, we have different pathogens associated with ESCA, and strains are slightly different. So we collected information, and CBS did this collection of data in the various countries. Then we developed a diagnostic tool. Vincenzo Mondello, who took the floor earlier on about GTDs, told you about a European project. It is the Microbial project that involves specific countries, and there an instrument, a tool, was developed for 11 types of fungi in order to be able to identify them in the nursery and have an adapted control action. Within the European project, we adopted the very same technique with 13 European countries. that decided to participate, 13 laboratories that were sent 12 samples of disease associated and non-associated with GTDs. So we had the controls. And we asked the laboratories to tell us what fungus was present in the various samples so as to validate the technique. That was done. The 13 laboratories found identical results. Now we have to communicate the technique so that it can be used in the nursery or in the vineyard. Another important point was already described by Vincenzo. Within the framework of the COST action, we synthesized the development cycle of these pathogens associated with the GTDs so as to communicate to European countries affected by the disease about this. And a number of members joined the action when we started it, but knowledge was very limited. So one of the goals was that of sharing knowledge and summarize it and spread it. So after 4 years of those countries that were lagging behind at the time in terms of knowledge have acquired a lot of knowledge. You can visit the COST Action website or Y Network for further information. And we put here also information about fungi and pathogens. So we, within the framework of the COST action, we sent a questionnaire to the partners with a reference person for each European country. We used the questionnaire to ask what if the diseases were present, which ones. We focused specifically on ESCA, then about Triosphera, dieback, dead arm, scoliosis, and blackfoot. This was done in 99 wine regions with table grape and wine grape productions, because depending on the country, these percentages change. We saw that the main cultivars involved are Sauvignon Blanc, Chardonnay, and Cabernet Sauvignon. We saw that the rootstocks associated are 4 in number. We identified 4, and as for pruning, it is especially cordon and guyot which are involved. The study was done on both young and adult vines. Oftentimes we hear that GTDs affect especially 8 to 25-year-old plants. This is what people said until a few years ago, because young vineyards were not studied. By now they have been studied, and we realize that also even 3, 4, 5 years after planting, you find GTDs. And so it was important to make those studies and do experimental tests also in young vineyards, along with old vineyards. It was difficult and would have been difficult to try and draw conclusions based on questionnaires, because you have different soils and cultural practices. And of course, all these factors, all these practices entail that the disease is more or less expressed, and that is also associated to the cultivar, to the variety. Parallel to that, we made another action about microbial ecology and did several samplings in several European countries. Due to the variety of the cultivars, we could not involve all varieties, but we took those grape varieties which are more present, which are Chardonnay and Cabernet Sauvignon. We used rootstock 5SO4, and we did so especially with a conventional training system with plots aged 15 to 25 years old, and studies were done. So the goal was that of characterizing the microflora and see whether in these European countries there was a common microflora or not, and specifically so a beneficial one. We know the pathogenic one. The pathogens associated with GTDs are present all over the world. Now the point is to see whether within the framework of that microflora, beneficial one, we have the same microorganisms all over Europe, and we could use them for control purposes. We want specifically to talk about the usage of trichoderma and the Bacillus subtilis partly. There are 3 microorganisms left, and this is very little when compared to the complexity of the diseases of the vine, because there are multiple pathogens. If we want to control them, clearly we have to have complex control approaches. And this will entail having growing practices and also biocontrol agents, including trichoderma. Bacillus, trichoderma are fine, the 3 microorganisms are fine, but we have to define this microflora and associate a complex of beneficial microorganisms, microflora, to all the rest. Then there is the interface between plant and pathogens, and with its interactions, there are several factors that need to be managed properly. We need control schemes which have to be simple, and one has been nicely characterized to study Euthepia, and then another one was done for ESCA, and a third model was developed to study pathogens which are associated to Botryopsphera dieback. An important element are the varieties, because some are more sensitive than others. We have to work on Chardonnay and Cabernet Sauvignon, which is more susceptible, more sensitive. The importance of the rootstock also needs to be recalled. Oftentimes, when you do nursery experiments, you totally forget the rootstock, while recent studies evidence that these can have a role in the expression of the disease. The COST action started in 2013. In the beginning, we had little information about associated fungi. And in 2014, everything has been sequenced, and that is important. Thanks to the genome sequencing of the fungi, we know what molecules are produced by the fungi. It can produce enzymes that degrade the trunk, and we have to investigate their diversity that causes necrosis in wood. We also know that fungi associated to GTDs can produce toxic molecules for the plant. Probably these toxic molecules, these agents, are involved in the leaf expression of the pathology. We need to distinguish between healthy carriers and those that are also symptomatic. And this has, of course, a repercussion on the productivity of the vineyard. In this interaction between plant and pathogens, as I told you, we also discussed extensively on a better characterization of the aggressivity of pathogens. They are present in the lignified part of the plant. A vine is diseased when it has foliar diseases, leaf symptoms. Pathogens evidently communicate at molecular level, and there is this communication between the fungus and the expression in the herbaceous parts where there is no pathogen. And this happens through molecules that go from the wood to the leaves, hence the need for these molecules. We need to identify them, and sequencing the fungi gives us insight in this respect. And then we have to know enzyme activity and characterize it, and that is associated to necrosis in wood. Many studies have been made on the impact of these studies on the physiology of the plant. First of all, in terms of a carbon mechanism, which is related to the vigor of the plant, and then defenses which can be induced to control the fungi. I told you that there are plants which have the fungus in the wood but do not have leaf symptoms. Now, this means that the plant controls the pathogens and controls the aggressivity, because it deploys defense actions in order to detoxicate the molecules produced by the fungus, and in order to keep the fungus at the center of the plant. And if we manage to control these elements, possibly we will also be able to stimulate them at strategic times when we know that the plant can become a diseased carrier after being healthy. Then we had a study in COST on nurseries, and this is the fourth axis. There are more than 666 nurseries associated in that, coming from 15 European countries. Main conclusions are there, but you can, of course, read the full article because it has been published. The point is optimizing the production of vegetal material in the nursery and in the stock and rootstock, and at the level of rehydration before and after storage. You also have indications about what is done at hydration before and after that as well. Here we have 52%, which is before cold storage. In those cases, there is no cleaning of the instruments, while the situation is totally different after, or before, rather, grafting, as the slide says. With reference to the disinfection of the pruning tools, we see that in 52% of cases they have not been disinfected. Of course, this does not contribute 100% to the expression of the disease, but it favors it. With reference to the feedback we received from the questionnaire, there was a question about means used to reduce the onset of pathogens in nurseries. You see that chemicals are used still. Not everybody uses them, but the usage of biological control agents is limited. Why? Because we realize that people told us that little is known about the beneficial effect of using BCAs. And if we then consider what happens in the nurseries, here we see the different moments when cuttings are produced and fungicides are used. With reference to BCA, what you have here is a very limited usage of these agents. The main ones are mentioned here, and specifically Trichoderma, Bacillus, and Glomus intraradices. We know that communication between scientists is not always excellent. Therefore, in Spain, a specific organism has been set up to favor communication, and this is open to nurseries and to other professions. This is the article that was published. You can read it if you want. It is available on the COST Action website. Disease management in the field. There is a slide about this, but this has already been extensively tackled, so I will not again repeat everything. I will simply say that we need to respect the plant architecture. We need to restore the rootstock and the density of plantation and so on and so forth. But all this has already been said. To conclude, within the framework of this project, which is going to be concluded in November, in July 2017 in Reims, we will have the 10th International Workshop of Grapevine Trunk Diseases, and it is an open event. You simply register, and every 2 years we meet, and scientists working on GTDs group from all over the world and spend 3.5 days talking about pathogens, their characterization, microbial ecology, the plant microorganism relations, and that in the nurseries and in the vineyard as well. Thank you for the attention. If you want to have more information about the COST Action, you see the website there and you can visit it. Thank you.